Modern Food Preservation Techniques by Nick Skillen. Electroporation and food irradiation. To preserve the safety and amount of food that's exported daily throughout the world, chemistry techniques are used to efficiently treat food and prevent it from spoiling. Modern nuclear chemistry and electrochemistry are used to kill bacteria and other microorganisms that cause the food to spoil. Electroporation and food irradiation are common methods to sterilize food for export and kill types of bacteria that include Listeria, E. coli, Campylobacter, and Salmonella that cause food to spoil. Electroporation. Electroporation is a food preservation technique used to kill bacteria and other microorganisms by sending a series of electron pulses through the food. Electrons are sent from electrodes in the device to kill the bacteria on the food by increasing pores in the cell membrane that cause a bacterial cell to burst and die. Electroporator devices control the size of the electron field based upon the cell type and whether or not the cell has a cell wall. Also, electroporators increase the concentration of electrons flowing throughout the field if more food needs to be sterilized. Food irradiation. Food irradiation is the process of exposing a food source to ionizing radiation using either X-rays, gamma rays, ultraviolet rays, or concentrated electrons to kill off microorganisms. Killing microorganisms prevents the spread of bacteria and reduces the spoiling of the food. Ionizing radiation is created by atoms concentrated in the form of waves. These waves are categorized on the electromagnetic spectrum from high frequency to low frequency. High frequency waves such as gamma rays, X-rays, or ultraviolet waves are more efficiently able to penetrate through substances such as food to rid the product of microorganisms that cause food spoiling. Irradiation techniques rely on three primary methods: electron beam generators, cobalt-60 gamma source, X-ray accelerators. Cobalt-60 gamma source. Cobalt-60 is a radioactive isotope used as a radiation source for food irradiation. This isotope emits ionizing radiation in the form of gamma rays, which are able to penetrate through most substances to sterilize the food source by killing all microorganisms and bacteria. The amount of time the food is exposed to the gamma radiation depends on the type of the food needing to be sterilized. Fruit and vegetable cells have cell walls, compared to meat, which has no cell walls. Therefore, more radiation is needed to sterilize plant cells versus animal cells. X-ray accelerators. X-rays are generated from linear particle accelerators that use a series of charges to give particles more energy and to become waves. Increasing the energy of the charges causes the frequency of the wave to increase. At 1 times 10 to the 16th hertz, the wave frequency turns into an X-ray. These X-rays are used as radiation to penetrate food substances and sterilize the product of microorganisms. A decrease in microorganisms helps prevent the food spoiling process. Electron beam generator. Electron beam generators form strong beams of electrons that ionize the product or take electrons away from the atom penetrated. This ionized environment is unsuitable for bacteria and other microorganisms to grow and helps prevent food from spoiling. In summary, electroporation and irradiation are in widespread use to help assist with food preservation and prevent outbreaks from bacteria-infested foods. Human advancement in the area of nuclear and electrochemistry provides many benefits to society. The prevention of food spoiling is one of the most practical day-to-day areas where we all benefit from these scientific discoveries. Citations.